Well, thank you for being present in this seminar. It is the third seminar of the Alumni of the Brain and Mind Sciences seminar series. Our speaker today is Gerasimos Gerardos. Gerardos has a degree in psychology from the Aristotle University of Thessaloniki. He graduated in 2019, and then he did his master in brain and mind sciences at the University of Crete, from where he graduated in 2024. At present, he is a PhD candidate in cognitive neuroscience at the University of Leipzig and the Max Planck Institute for Human Cognitive and Brain Sciences, His thesis is on the verbal working memory with concurrent transcranial direct current stimulation and functional magnetic resonance imaging. And this will be the topic of his talk today. Gerasimos, thank you for accepting the invitation. The floor is yours.
Thank you very much for inviting me today. It's an honor to present to you guys the field of work I'm doing. And thank you for the very kind introduction. Yeah, my first degree was in psychology. And I think since then I was always interested in the brain and how psychology relates to the brain. And that led me, when I finished my bachelor's degree, um, um, to come to the brain and mind master's program. What really I found like very inviting on this program when I was searching, it was of course that it was a very high quality program with a lot of that would give you a lot of practical experience through rotation. Like 1 year of labs is something hard to find, guaranteed in a master's program. And I think the main selling point was that the program was interdisciplinary. So a lot people from a lot of different fields could come together and share combined knowledge and views of the world. Um, of course, when I started with the program in 2020, it was the beginning of COVID, or like in the midst of COVID. So life in Crete was a bit weird, and the program was not, I think, in its full potential in the beginning, being online. But I think everyone made their best effort to give high quality teaching. Um, and I think that this interdisciplinary approach was the difficult part about the program. As a psychologist, I had all the theory in my mind and how things might work, but it was theory without really the biological basis of it. Or the computational, let's say, modeling of these phenomena. So I think integrating all of these can give you a unique way of viewing the neuroscience world, let's say. And I'm very thankful for that. I think it made me a more complete scientist because with only theory, you can dream big, but like with also the biological reality, you can dream like for real things to happen and for real things to find. Um, so after finishing my master's, actually, while I was doing my master's, I decided to do an Erasmus internship, which is extra to the curriculum, just to see how the science world is outside of Crete. And that led me to the Max Planck Institute for Human Cognitive and Brain Sciences, where I did approximately 1 year of rotations there. And this is why I graduated on 2024, because I wanted to retain my student status so I could be a student assistant here. And when the time came, with a lot of hard work, I would say, and patience, I managed to get a PhD position in the group I was being an intern. And um, today I'm going to present you some work that we're doing in the lab and the main findings that we have so far from my PhD thesis. Um, yeah, of course, what I would advise to everyone in the all the students that are now in the Brain and Mind program is to not be afraid to take risks, prolong your studies, do things that are extracurricular, that are not like mandatory, because this is where the real juice of science is, the extra stuff. And um, human cognitive neuroscience is not fully covered, or it's not the main topic of the program, but there are many labs out there in Europe and beyond that do this stuff, and no one would say no to you to go and experience something more than what is offered. So that would be my advice. Um, so I think I talked quite a lot about myself and about all this stuff. So I'm gonna begin with my presentation. So in my presentation, I'm gonna talk a bit about the theory or the field behind human cognitive neuroscience, what exactly it is, and I'm gonna present to you the main methods that human cognitive neuroscience uses, so that you have an idea of what can be done studying directly the human brain. Okay. So, what is cognitive neuroscience? So, it is the field that examines mental processes and how they emerge from brain activity, and they end up giving you some sort of behavior. Of course, this is not a linear process. All these three interact, and it is very hard to know or to say, or maybe it's something of the work of a philosopher to say, like, where everything starts. Is it the brain? Is it actual behavior? Um, but yeah, it is a field that combines all of these fields of study and phenomena, and combines it and studies it all together on human brain. So the basic methods of cognitive neuroscience is studying behavior, so behavioral tasks, using neuroimaging, so ways of seeing brain activity, and neurostimulation, which is a way of probing the brain to cause changes in activity in the brain. Of course, when you're designing a task to use in one of your experiments to see and measure an ability or a cognitive process, you have to have the concept in mind, for example, working memory, and then you have to define it in very specific terms. How do you use this term in your study? And usually tasks give you this operational definition, as we say. And it's your work as a scientist to create a task that isolates the very specific, let's say, ability that you want to study, and then also design a control task, which is a task that, let's say, cleans what is embedded in the isolating task, your central task, meaning that a task will check about for a lot of abilities, and you want to cancel out the abilities you're not interested from this task by designing another task that does not contain your main ability or yeah. So the main neuroimaging techniques that we use in human cognitive neuroscience are, of course, non-invasive ways of measuring the brain, and it's either magnetic resonance imaging or EEG, so electrical signals of the brain. I will talk more about them in a bit, of what exactly they measure. And of course, we have a lot of ways of stimulating the human brain without having to make any surgeries or drill any holes to put any electrodes. So we can use magnetic fields and cause changes on the electrical fields of the neurons inside your brain. We can also apply direct electrical currents, or even more new techniques, more fancy techniques. You can cause changes in brain activity by infrared emitting light, which causes changes in the metabolism of neurons. And of course, using the mechanoreceptors that all neurons have, or most neurons have, you can also cause changes in activity by ultrasound stimulation. Of course, it's not as defined or as focal as one would like, because you apply it over a big area of the brain, and it's not a small electrode that you put in the brain, but it still gives you a way to change the activity of the brain and notice differences on task performance or in brain activity. So what is really magnetic resonance imaging? So you want to measure the brain, and you can measure the structure of the brain or the functional properties of the brain. And what you use is actually a big magnet, and this magnet has an effect on the molecules from which everything is made of. So the protons. These protons have a specific spin. So imagine it's a ball that turns around itself. What you do is you align with a big magnetic field all these molecules of the brain, and then with radio frequency currents, you change the direction of this spin, and then you turn these currents off. So these protons are going back to the aligned magnetic field. So why this gives us information for the brain? It is because different material, different chemical composition of matter, like axons of the neurons, or the somata of the neurons, they react differently to these changes of the magnetic field. So, for example, I don't know if this video can play. Actually, it cannot, but it was a very good illustration of what I'm talking about right now. Let's see, Anna, here we can play it. So you can see that you apply on the spinning proton a magnetic field. It aligns with this magnetic field of your scanner, and then you bombard it with some radio frequency currents, and that causes changes on how it spins. As soon as you turn it off, then your proton releases energy and goes back to the original position, and what you actually measure is this energy. Okay. So how do we measure the function of the brain activity itself? So you don't actually measure brain activity as electrical signals as we know it from systems neuroscience or from biological neuroscience, but you measured it as changes in the blood flow and in the oxygenation of the blood that comes inside your brain. So there is a great coupling of how your vascular systems react to when neurons are active. They have increased metabolic demands, and that causes increased metabolic demands, and through the glial cells, the blood vessels inside your brain can get the information that they need more blood. So actually you can measure how much more oxygen is coming within the brain, and this correlates very nicely with electrical activity, like with excitatory postsynaptic currents in the brain or with LFPs. So it's a very validated method of measuring brain activity. And a little bit more about the brain stimulation techniques and how they work. So it's not really magic, it's just physics and chemistry. You apply a magnetic field through a magnetic coil on the brain, on the area that you want to stimulate, and then it causes changes in the brain activity. And you can simulate or expect what kind of changes you will have under this magnetic pulse, which translates to brain activity in the neurons, especially those on the cortex that have that are perpendicular to the surface of the coil, and that can cause even behavior like muscle twitching or movement of the hands. So it sends a signal that actually travels through, for example, your spine, from your motor cortex to your spine, and causes movement of your hand. And of course, this is a very simple example, but you can also apply this on areas where you believe that cognitive functions are being served. And of course, that would mean seeing changes on behavior, on memory, for example. And another way of doing non-invasive brain stimulation is applying the electrical stimulation. So you either apply a small direct current, which is stable through time, or alternating current to take advantage of frequency-specific rhythms, let's say, of the brain and of cognitive processes. So what is done, what is actually, how it actually works is that you create a sub-threshold depolarization on the anode, so the positive electrode. So cells there are more active, or they're more prone to be activated, because you don't cause spikes here as you do with TMS, but you only make your cells more excitable. And of course, on the other side of the stimulation, the cathodes, which are the negative electrodes, you create slight hyperpolarization and make cells more difficult to spike. And of course, there are different montages of how you put electrodes and different kinds of electrodes. You have these crude standard approach with huge electrodes and no spatial specificity, or this circular montage where you stimulate the central part and you, let's say, try to avoid spread of this stimulation to the surrounding tissue. So, of course, again, cells are not really like aligned, all of them in a perfect way. So, of course, it's not 100% all the cells under the anode will be stimulated or positively or negatively. So it really depends on how they are aligned. But research shows that it works, and you actually have this increased activation under these spots and even behavioral changes. So all of these have to be combined to do good research, let's say, into, or the best possible research in a holistic approach. So you want to use all these methods together so that you can apply stimulation and simultaneously record the brain and see what really changes in local activity where you stimulate, what it changes in the whole network activity, and how does this change the cognitive processes, which in the end are measured by behavioral responses of some sort. So that's some basic information about how human cognitive neuroscience works and what the methods are that they are used. And now I will present some examples of how all of these things can be implemented and produce some sort of scientific knowledge. So I will present some analysis that we did for my PhD project, which is exploring verbal working memory with focalized tDCS. This is done with collaboration of the Cognition and Plasticity group in the Max Planck Institute, and in the BioCog group, biological and cognitive psychology in the University of Leipzig. And of course, it is part of a bigger interinstitutional project, which is done all around Germany. We all use the same approach and the same methods, and we use tDCS simultaneously with fMRI to study human learning and memory in different functional domains across the lifespan. So we're currently during the first part of the project where we test people which are, let's say, young adults from the age of 18 to the age of 45. The second part of the project is gonna be implemented later, and it's people above 45 years old and with some sort of mild cognitive impairment. So every different institute has chosen, let's say, a different type of ability to test, and so we have a holistic, let's say, approach of learning and memory, and we are testing verbal working memory here in Leipzig. So I'm going to present only things that we do here in-house. So the basic study design is that we use fMRI, tDCS, and a task measuring working memory all together inside the scanner. It is a very complex study design. It tries to tackle the problem of low samples in human cognitive neuroscience. So we're trying to gather up to 100 people of this age range. And we have actually, we invite them 6 times to do different experiments. In the beginning, they all come for a neuropsychological battery so that we can test their overall cognitive level, let's say. And we invite them also at a baseline scanning session where we measure a lot of where we gather a lot of structural data so that we can know or relate inter-individual differences between brains. And of course, then we have different groups of applying tDCS. The first group that is already completed is the sham group. So this was more like a piloting phase where we gathered fMRI data with fake stimulation. And then we have the active groups and in the target and with stimulation on the target side, and the active groups with stimulation on the control side. So we are testing for the left stimulation on left inferior frontal gyrus, which is very much connected with verbal working memory and executive functions, and the right occipital temporal cortex, which is less related with verbal working memory. Of course, it is connected a lot with attention and spatial awareness, spatial attention. So, of course, for the active groups, we do control for stimulation. So we don't only deliver, let's say, real stimulation, but we also deliver fake stimulation so that we can check what brain activity is specific to stimulation and not noise or random effects. Today, I'm going to only present data, unfortunately, from the sham group, from the fake stimulation group, because the active groups are still underway and we are blinded. So it's a double-blinded study, so no one knows except the people that handle the stimulation what kind of stimulation is delivered at the participants. I'm going to present to you three analyses. The one is a behavioral analysis, the other one is a functional fMRI analysis, and the last one will be a structural fMRI analysis. So the first work is actually part of a bachelor thesis by Marie Böttcher, and it is application of principal component analysis on our neuropsychological test battery and how it correlates with the task that we chose to measure working memory with, which is the NBAC task. So we're going to talk a lot about executive functions, and they can be viewed, or we can define them for the purpose of this talk, as general purpose mental processes that regulate overall cognition and behavior. So it's multidimensional, it has different facets, and one could focus more on three of them, which is inhibition, so being able to suppress stimuli and select the ones that matter. Shifting, which is viewed as cognitive flexibility. It means that you can change tasks you are doing and control the demands, your cognitive demands that you are using to do different tasks. And of course, we have updating. So this system is not, it's dynamic, it always changes, and you have to update all your resources and all your, let's say, facts about the world that you know at this specific point. And of course, our main focus is on working memory, which is defined as first the ability to maintain and manipulate information in your mind. It is an ability that is embedded in a lot of cognitive functions. So you need it for learning, you need it for retrieving memories, you need it for attention or for internally guided behavior, like goal-directed behavior. And of course, it is necessary for the perception of time. So without working memory, we would only be able to experience the world as it is in every second and not remember what happened the previous second. And it creates this, let's say, continuous experience of the world. Without memory, we cannot really have this continuous experience. Of course, there are different ways of, let's say, conceptualizing working memory. I'm not going to go too much into this for the sake of time. And I will move now on the specific task we use to measure working memory, verbal working memory. So it is the NBAC task, and it's a letter NBAC task. In this task, letters appear on your screen one by one, and you have to press a button on different occasions, depending on which is the rule, which is the type of task you're doing, the condition of the task. We use three different conditions. It's the zero back, so you just have to attend and press a button whenever an X appears on your screen. So little to no memory is required. And then we incrementally increase the memory demand. So you have to remember the previous letter or two letters before or three letters before. And this is an important design to be able to actually check if what we measure is actually sensitive to these demands. So the more the demands, the more difficult the task. And to assess it, we use sensitivity, which is D prime. It is a very nice measure because when you have to press or not press a button, depending on whether you have a matching letter or a not matching letter, then it's not only being correct or wrong. You can press the button when you shouldn't, or not press the button when you should have. So this measure, let's say, combines all of these possibilities and probabilities to create one single measure. And it can also measure your response bias in a way that some people tend to withhold their pressing and be more conservative if they feel that they're not sure, or some people just press a lot. So you can delineate sensitivity, so the ability of people to detect certain stimuli, from their response bias. And what we found is that definitely our task can successfully grasp these changes, both in terms of sensitivity and of reaction time. So the behavioral performance systematically declined with increasing memory load. And then we applied on that principal component analysis. So why did we do that? It is one way to theorize and use let's say, research to say, okay, my task measures actually working memory. But if you want to be completely sure, you want to use other tests and see that, let's say, check the same kind of abilities in different ways and see, is my task actually related with all these tasks? And one could go about it doing just correlations, but this is not an efficient way. And also there's a lot of chance that you get spurious correlations. So what we did is we applied principal component analysis. In few words, principal component analysis tries to find latent variables, hidden variables within a multidimensional space of data. Imagine your neuropsychological test being a dimension of your data, and then you have, let's say, 8 or 10 or 20 of these tests, and you want to find three main components that can summarize abilities tested by all of these correlated tests. So the tests that we are used are here. I'm not going to go into too much detail about that, but they were measuring different parts of working memory, of long-term memory, and of executive functions like cognitive flexibility or inhibitory control. And we ended up with 4 different principal components. So we could summarize it into 4 different variables. Of course, this analysis produces as many variables as your original data, and it's up to you as a researcher to choose how many are enough to describe the main essence of your data. We decided that 4 was a good number based on various criteria that is not very important to talk about at this point. But what we found is that there were 4 underlying variables which we named based on which tests loaded greatly on them, as executive flexibility, verbal working memory capacity, visuospatial recall memory, and verbal recall memory. So this is more long-term memory, and this is more about short-term memory, or just a general executive ability. So general cognitive control. And we wanted to see how our NBAC task that I described correlated with these different dimensions of the data. And what we found is that it greatly related with executive flexibility rather than working memory capacity. Of course, it related with both of them. So we do know that it does measure changes in the load of your memory. How good are you at retaining things at your short-term memory? But also it focuses more on the manipulation of these data, how good you are at analyzing or using this information. So having, let's say, validated that our task is good to go and we can measure working memory and much more the executive aspect of it, we decided to look at the functional data of the brain. And this is a paper that is going to be soon out and published in iScience. And it actually applies dynamic causal modeling to test how different nodes in the brain causally interact with each other. So, of course, how do we get brain activity from all that we said before, from the blood response? So we apply something called the general linear model in the hemodynamic response. Hemodynamic response is the blood changes because of events that happen, like stimuli coming or you doing a task. And it looks kind like that. So blood flow and oxygen increases when you see something. And you can apply this model to your own task design. So we deliver the task in blocks. So for around 50 seconds, people are pressing buttons on different conditions, 0 back, 1, 2, or 3 back. And you can model this like that, like a hemodynamic response that lasts as long as your task block. And of course, what GLM does is it takes what you have as a signal from the MRI and it fits this model. And what you actually get is how well it fits in the form of beta values in this, let's say, linear equation. So it's linear terms with weights with a bit of noise. Yeah. And in order to test how specific conditions are represented in the brain, you want to contrast conditions with each other. So if you want to get, for example, the specific activation of the 3-back task, of the high demand task, you can, let's say, do that activation minus the activation that you get from the 1-back task or from the 0-back task. And in this way, and with a lot of statistics, you end up with a map looking like this, with areas that get more blood flow, so you suspect that they are more activated during your task, and areas that are less activated during the task, statistically significant. And from that, you can depart and with some extra analysis, draw regions of interests. So how different regions are organized and utilized in your task. And of course, what we see in this is that working memory or the NBAC task really utilizes the multiple demand network, which is a network that is used in general when you do anything cognitive. And downregulates in a way the default mode network, which is the networks that are activated when you do nothing, when you just daydream, when you are more like internal and you just let your thoughts loose. And of course, when you have these many regions of interest, it's very difficult to do any statistical test. So you have to decide on which regions are of your own interest, on what you want to test. And you inform yourself usually on choosing based on previous research done. So what we did is we chose bilateral IFG, inferior frontal gyrus, inferior parietal lobule, and the top part, the superior part of the cerebellum. And we did that because we were interested on what is suspected that these areas are connected to as in terms of abilities. So IFG is connected with verbal working memory, and it's suspected to be where articulatory rehearsal and executive control is happening. Of course, not really happening only there, but it plays a significant role on these processes. The inferior parietal lobe in animal research and also in human research has been connected with phonological storage or maintenance of information, or where information from different modalities merge. And of course, the cerebellum is an area that not a lot of research has been done yet. So we really wanted to see how it's connected with our data. And of course, it is connected with prediction forward modeling. So what, let's say, it's expected to be seen or be done, and with error monitoring of action. And of course, we wanted to validate that these areas are indeed responsive to our task. And we saw that, yes, indeed, activation increases as task demand increases. And of course, with a ceiling effect when the task becomes very difficult. And to test the relationship between these 6 areas, we applied dynamic causal modeling. So in a few words, dynamic causal modeling views the brain as a dynamic system of regions interacting with each other in a causal manner. So activity in one region drives activity in another. And what it aims to do is to take into account experimental inputs as conditions or specific tasks, to see how these change the interactions between regions of the brain. And of course, in simple terms, what it does, it takes a plausible network architecture. What do you expect to happen? And then you run simulations that say, okay, if I have these expectations, can my simulation with this model that I created predict the fMRI signal that I gathered? And then you slowly, in an iterative way, you change the model and you change the connection weights between the areas, and you only stop when a lot of the variance of the data is explained by your model prediction. And after you do that for every subject, you want to do it in a group level, and you want to find common patterns and explain differences by group information, such as conditions or which group your participants belong to. And you want to find the connections that are really important and remove any connections that are not reliable or do not give you any more prediction value on your model. And what you actually get out of it is 3 terms which are important. The intrinsic connections, so how your areas interact in a baseline state, or let's say when you don't, for us, it was not just a baseline state, it was a general task state. So when you're not having the different demands and it's just all the tasks, what does the architecture of your network look like? And then you have modulations, which is how tasks or task conditions modulate this baseline general state, and a driving input, which is where does the task enter the system? So where is the task first, let's say, changes activity, so you can have the cascade of the task network. And what we found is that if you initialize your model with all possible possibilities of inputs or strengths or modulations or interactions, then you end up with a very complex architecture which can be let's say, summarized in three terms. So left IFG is your entry point. This is where the task starts affecting the brain. And then you have in the baseline state, in the general task state, bidirectional frontoparietal cerebellar architecture. So all the areas have some sort of interaction with each other, with the parietal control being inhibitory. So all the connections are positive and facilitating. So one area facilitates the other, but the parietal cortex seems to inhibit all the other areas. So it gives us information about the parietal cortex being more of a regulator of activity, and the other one more driving the processes. And of course, how does the different tasks, conditions like the one back, the two back, and the three back change this architecture? And this can be summarized here. You see that this baseline states in the one back changes by the feed forward, let's say, excitation or facilitation of the parietal cortex from the left IFG. As task demands increase, the architecture changes completely. It is noticeable that the communication between the parietal cortexes become null, so they stop talking, let's say, to each other. And then you have increased facilitation from the left IFG and decreased inhibition from the parietal cortexes. And you see here the cerebellum starts talking less to the other areas. So as task demands rise, the cerebellum is not much implicated in the cognitive process of working memory. When you reach very high demands, you see that the crosstalk between the left IFG and both the parietal cortexes become heightened, they are positively modulated, and they facilitate now each other. So we go from, let's say, inhibition to facilitation. So what do we take from this? So increasing working memory load is led by strong task modulation. The frontoparietal connectivity is strengthened, and the cerebellar connectivity is weakened. So in order to do more intensive tasks of working memory, you don't have, let's say, enough time for error control or for feed-forward models. You have to focus all your energy and brain demands on the cortex. This is our take on that. Of course, interpretation is always open. Then we want to see, is there a brain behavior correlation? So of course you see all these network dynamics changes, but that doesn't guarantee that individual performance can be explained by these changes. What we found is that indeed we have significant correlations between behavior and the effective connectivity, causal connectivity in a way. So the most important connections, as we see, is the left IPL, left parietal cortex, sending information or facilitating the left inferior frontal gyrus. So the more this connection is modulated by the task, the less well people tend to do the task. And the same stands as for baseline connectivity between the left IFG and the right IFG. How can this be interpreted is that when you're good at a task, you don't need as many demands, and you don't need as much changes on your baseline state. So it's more about efficiency of the network. Less energy means better performance. Here we applied Bayesian correlations. These kinds of correlations are very good and sensitive for small samples, where the frequentist approach, like Pearson or Spearman, cannot really give you much information. Here, because it's only a part of the sample, we only have 20 subjects, and it's not enough to capture any behavioral effects. Last but not least, we have the structural information of the brain, which blends it all together. This is a work by a master thesis student for the master thesis of Franka Hüttenheim, which is now being written. And it's actually connecting the microstructural foundations of the brain with verbal working memory. So, of course, you can not only get functional information about the brain with brain imaging, but you can also get structural information like gray matter, white matter, and cerebrospinal fluid. And you can segment the brain into areas that are more probable to be white matter, gray matter, and CSF. But you can also have a type of structural imaging which is called diffusion imaging. This kind of imaging takes advantage into how water molecules behave when they are in different amounts and in different, let's say, when they have different amount of space to move. And so we use sequences inside the MRI scanner that are sensitive on the changes of the on the water molecule movements. And we use a very new method, which is called neurite orientation, dispersion, and density imaging, which can actually create models and, let's say, give you an idea of different aspects of the tissue underlying the brain. And it uses by delineating intracellular, extracellular, and CSF, meaning free water diffusion of water particles. And in simple terms, you get two, let's say, the most important indices that you take are information about how aligned your fibers are in the brain or in the underlying voxel of the brain, the parcel of your brain you're looking at, and how dense your neurites, actions, and dendrites are in this. So alignment and density. So are there many axons and dendrites, and are they aligned or not? And this is actually validated in animal models or post-mortem human models. So it's actually pretty valid. And what we found applying these metrics on the same regions of interest that we had for our functional data is that although we have a lot of variability between people on these indices, on the neural density index, only the left IFG was the one that was correlated with task performance. And not only that, but we had to take only the gray matter of this area to remove the noise of the other kind of tissue. So this metric is also tissue specific. It is different things happen on the gray matter with neurite density, and different things in the white matter. And it seems that when it comes to density in the left IFG, what matters for performance is the gray matter. And of course, we did the same thing for the orientation dispersion index, so how much aligned these fibers on the underlying tissue is. And we found that in general, all areas had somehow like a weak correlation with individual performances, more in the anecdotal side, but still giving a sense that also orientation dispersion index has something to do with performance. I have to remind you that our sample is still very small, and we will do that for 100 subjects. So we expect to find stronger results, but that's, let's say, a nudge towards where the data is going. And overall, what we find from this study is that both these indices, both the alignment and the neurite density has a negative correlation with performance, taking us more to the neural efficiency hypothesis. So I remind you that the less ODI is, the more aligned the axons are, and the less NDI is, the less neurite you have, like less in dense neurons you have in this voxel, in this area. So that means that people that have less dense left IFG seems to, in their gray matter, seems to perform better. And people who have more aligned axons in gray matter and white matter also seem to perform better. So again, that tells us probably there is it's neural efficiency again. You don't want dispersed connections that go everywhere. You want very specific connections and not too many. More like how synaptic pruning works. So you want only the connections that are worth it for your task. So this was all the, let's say, initial results from my PhD project. And we see very nicely that you can get from human brain cognitive neuroscience techniques and methods different information about the brain. Structural, functional, and also behaviorally related information. What we want to do continuing this project is we want to integrate these different modalities into one model, not view them separately. We want to link brain and behavior and use all of that to, in the end, understand the transcranial direct current stimulation mechanisms. See, does actually stimulation that we are applying change network dynamic? And is this stimulation more efficient depending on the state of the brain or the structural underpinnings of the area we're stimulating? And of course, all of that aims on giving us personalized interventions. Use these stimulation techniques to improve working memory in people with dementia or with mild cognitive impairment, or even generalized in other domains like depression or either types of psychopathology where working memory is a basic thing that gets affected. And this is all the references where you can find the things I talked about. And thank you very much for your attention. And I'm open to questions. I also want to thank my team at the Max Planck Institute and at the University of Leipzig for making this project possible. Thank you very much.